In the previous lesson, we learned about innate immunity, the importance of skin and the mucosa, and how they block invading pathogens. How macrophages, along with neutrophils, combat enemy microorganisms that have breached our first line of defense. Macrophages are armed with PRRs, receptors that recognize structural patterns, PAMPs, that distinguish foreign threats from self, the components of our cells in our body, and non-self. Finally, we found out that not all bacteria are bad, and that our microbiome actually partakes in fending off unwanted intruders. When innate immunity is overwhelmed, however, and unable to successfully repel intruders, adaptive immunity kicks in. Two major aspects of adaptive immunity make it particularly effective and very different from innate immunity. First, the adaptive immune system is exquisitely specific. Its cells, the lymphocytes, are armed with sophisticated receptors that are able to focus and discern the finest structural details that precisely discriminate between us, that is, self, and pathogens, non-self. This unique capability allows the lymphocytes of adaptive immunity to get rid of the enemy with minimal collateral damage. Second is the fact that once T and B cells have learned how to best knock out a particular threat, they deposit memory cells in the lymphocyte archive. These memory cells are called upon in the event a pathogen attempts to infect us a second time. In this case, the pathogen is met with an immediate and strong response, clearing the threat before it can take hold and cause disease. So basically, the unique strength of the adaptive immune system is its memory and the ability to recall T and B cells armed with receptors, sophisticated, fine-tuned precision sensors. The B cell receptor, the BCR, contains a membrane-bound antibody. Antibodies, also called immunoglobulins, are relatively large proteins made up of 4 subunits, 2 heavy chains and 2 light chains, arranged in a Y-like shape. The 2 tips, where the heavy and the light chains meet, form the binding sites that recognize the antigen. Consequently, a single antibody has 2 identical binding sites. The stalk of the Y, where the 2 heavy chains meet, forms an effector signal. And as we shall see, this too is very important. The binding sites of an antibody recognize very specific yet limited structures of an antigen. These sites are called epitopes. Now, metaphorically of course, imagine an antibody whose epitope corresponds to the very tips of our fingers. This antibody would clearly read the details of our fingerprints, and although the binding is to a limited surface only, the antibody would be able to easily discriminate people with precision. Now, getting back to the BCR, the B cell receptor. The B cell receptor corresponds to the specific antibody that this particular B cell will ultimately produce and circulate in our blood. Each B cell can produce thousands of copies of its single one kind of antibody. Collectively, however, the billions of different B cells of our bodies can produce a vast variety of antibodies that can recognize every possible epitope in nature. When a pathogen encounters a B cell that just happens to have a receptor that can specifically bind one of its antigens, the system goes wild. This precise binding event triggers a process of clonal expansion. The specific bound B cell divides over and over again, generating thousands of B cells, all members of the relevant B cell clones specific for the pathogen. These activated B cells develop to produce and secrete millions of their specific antibodies. These will ultimately bind to the pathogen and neutralize it. Now, it turns out that antibodies can come in a number of different flavors and sizes and are adapted to specific tasks. Remember, in the very first lesson, we emphasized that life is all about structure and function. Antibodies have different structures, each with its special function. The very first type of antibody to be produced in response to the initial encounter of a B cell with a pathogen is called immunoglobulin M, or simply IgM. IgM has a binding site that recognizes its epitope with modest affinity. In order to compensate for this lower activity, however, IgM appears in the bloodstream as a complex of 5 copies of the antibody linked together to form a pentamer. Thus, although each IgM binding site may only loosely bind its epitope, together the pentamer's 10 binding sites work pretty well. Once the B cell binds its target antigen, the enemy, it can begin a course of basic training and learn how to best subdue the pathogen. This can take a few weeks. Through a process of trial and error, the genes that code for the IgM undergo local mutations and variations that refine and improve the binding of that antibody for its epitope. This process optimizes the binding site to become a super binder, which snugly fits its target, latching onto its epitope with tenacity and doesn't let go. In addition, the antibody itself converts to a lean and mean immunoglobulin called IgG. IgG antibodies are more effective. They live longer in our bloodstream and are quicker to bind and knock out pathogens, as we shall see. Another type of immunoglobulin are the IgA antibodies, which are especially adapted to survey and patrol our mucosa. IgAs are thus often the first antibodies to encounter pathogens and play an important role in clearing viruses that infect our gut, our lungs, and the urogenital tract. Finally, IgE antibodies are special antibodies designed to knock out and kill parasites. So as you see, the first B cell response produces large and somewhat clumsy IgM antibodies, but after a few weeks of training and maturation, antibodies become sophisticated guided missiles that intercept viruses before they infect our cells. Is this, however, enough to take out the enemy? Can bind specific epitopes of the... Think about it. B cells, armed with B cell receptors, can bind specific epitopes of the pathogen. This triggers that B cell to divide, to engage in a process of clonal expansion. Thousands of progeny, daughter cells, are produced exact copies of the mother cell. Each B cell produces antibodies that specifically correspond to the pathogen's epitope that stimulated the original B cell in the first place. Okay, but how, you may ask, does this help us fight infection? You surely recall that viruses are decorated with spike proteins, and that these spikes must bind to their receptors on the surface of the target cell in order to mediate cell entry. Only then is a virus actually able to infect us and begin to replicate. So now consider a B cell whose B cell receptor just happens to correspond to the viral spike epitope. And even more, specifically to precisely that part of the spike responsible for binding to its cell surface receptor. The encounter of such a B cell with its virus initiates clonal expansion, and now thousands of daughter cells begin to produce specific antibodies that target the viral spike protein. The viruses are attacked, first, with IgM, but by the end of a few weeks of training, optimized IgG-guided missiles are produced. These latch onto the spikes, intercepting the viruses and blocking them from binding to the cells of our body. Binding of antibodies to spike proteins can neutralize the virus and prevent infectious disease from the very get-go. Thanks to immunological memory, should the same virus attempt to attack us a second time, the optimized, ready-to-go trained B cells stored in our memory ignite a powerful, focused response to knock out the virus before disease has a chance to set in. This ability to recall is the basis of vaccination, as we will soon learn. Now, to be honest, viruses are not wimps and have some tricks of their own, as we will also see in the next unit. Some viruses mutate and modify the structure of their epitopes. In this way, they effectively disguise themselves so to become less recognized by the antibodies that trained to bind the original viral features in the first place. Fortunately, antibodies have some flexibility in accommodating to change and variation in the targets they bind. Hence, an antibody designed and perfected to bind a specific epitope can cross-react and bind variations of that epitope, but pay a price of lesser efficiency. Nonetheless, cross-reactive binding is often enough to halt infection of a virus that is mutated. So you see, there is a dynamic dialogue between our immune system and the viruses they attack. B cells mount perfected antibody responses to block invading viruses. Viruses under attack accumulate mutations in an attempt to masquerade their epitopes and evade immune attack. Thanks to cross-reactivity, antibodies can nonetheless use first-generation antibodies to fend off evading viruses until a second round of antibody production takes over. Antibodies also assist in combating bacteria. As you recall, resident macrophages eat to defeat. Macrophages devour bacteria that invade our bodies. Some bacteria, however, tend to be slippery and hard to catch and hard to hold on to. If these bacteria, however, encounter B cells, they can launch clonal expansion and the production of antibodies specific for the slippery, coated bacterial surface. When antibodies bind and cover such bacteria with antibodies, the stalk effector signals are presented outward. These stalks are recognized by the macrophage, who has special effector signal receptors on its surface. Binding the antibody effector signals by macrophages gives them something to latch onto and increases their efficiency in eating. Coating a donut with chocolate sprinkles enhances our eating experience. So antibodies can neutralize viruses, prevent disease, and assist macrophages in dealing with bacteria. However, in all of these encounters, the pathogens, viruses, or bacteria interact directly with the B cell to stimulate clonal expansion and antibody production. But how does the immune system cope with viruses that have succeeded to infect our cells and begin to replicate? Here is where T cells make all the difference.